We'll start soon.
Okay, we'll get started.
I think so. Yeah, let's
Okay,
do that.
great. Okay, good evening, everyone. So, welcome back to the another departmental video, another departmental webinar. And today we are pleased to have Dr. Vishal Rai, own alumnus, with us, and he's associate professor and Swarna Jayanti fellow at Department of Chemistry, Indian Institute of Science, Education and Research, Bhopal. He did his PhD from our own department from 2003 to 2008. He worked with Professor Nambudri. Subsequently, he went on to do postdoctoral. He had a postdoctoral position as well as MITACS Elevate Fellow position at the University of Toronto, Canada, before joining as assistant professor in Department of Chemistry, IISER, Bhopal in 2011. His research group mainly works on development of chemical technologies for precision engineering of proteins, and also they involve in synthesizing homogeneous antibody-drug conjugates, protein immobilization, and also he makes tools for peptide and protein synthesis. He is also a founder and director of Tech Private Limited, and it mainly deals with protein engineering technology, and he has several honors and awards on his name. He is a recipient of Swarna Jayanti Fellowship and also a Ramanujan Fellowship. And he is also a DAE Young. He also received DAE Young Scientist Award. And recently, he joined ACS Chemical Biology team as an early career board member. So today, he's going to talk to us about organic chemistry with protein enabling biology and medicine. Professor Rai.
Thank you, Professor Leela, for very kind and generous words of introduction. And also, I would like to thank the Department of Chemistry at IIT Bombay for giving me this opportunity. It's always good to be back in my own department. Now, for those who have not visited IISER Bhopal, you can see the main entrance over here. And in the background, even though we do not have Powai Lake and Bihar Lake close to us, we definitely have Upper Lake, which is right here. It's around 30 kilometers long and flanked by Shamla Hills. Okay, so getting back to the campus, our group at IISER Bhopal is developing chemical technologies for precision engineering of proteins. Besides, we are looking into how it can impact various technological demands of biology and medicine. But before I get into those details, let me tell you why such technologies are absolutely essential in a broader perspective. So the basic knowledge of reactivity and diverse aspects of selectivity is really essential for meeting the technological demands for the tool development for biophysical and biochemistry. And if you look into how the non-selective tools have captured the market, it really tells you the potential opportunity that lies ahead for the selective technologies. But what really excites our group in particular is the potential to contribute to directed cancer chemotherapeutics, particularly by synthesizing homogeneous antibody-drug conjugates or ADCs. Now, this is a state-of-the-art technology, and as the name suggests, it's a combination of a monoclonal antibody that is covalently linked to a drug or a toxic molecule through a linker. So now when these ADCs are given to a patient, the job of this monoclonal antibody is to bind specifically to an antigen that is overexpressed in the tumor cells, but not in the normal cells. And that's the basis of directed therapeutics. And this, after a while, this overall complex of ADC and antigen slowly gets internalized, and then it reaches lysosome, where the lysosomal degradation releases the toxins, which eventually leads to the death of the cell or apoptosis. So based on this platform, there are already a few drugs that have received FDA approval and have been serving in the market for last few years. And if you look into them, there's one thing common about all the drugs that are out there, that they are sold as heterogeneous mixtures. And what that means is that there will be 0 to n number of toxins attached per molecule of the monoclonal antibody. Now, this heterogeneity is definitely detrimental to the pharmacokinetics, and it also brings in batch-to-batch variability. But as you would anticipate, not much can be done about it due to lack of enabling technologies. And in the latter part of my talk, I'll be telling you about some of these stories where we have been able to contribute in this area. But while we are working on this protein-based therapeutics, we also realize that in the long run, such biologics would not be accessible to a huge population across the globe simply due to the cost factors. Now, that is something that is worth considering. And in fact, that's the reason why in long run, we want to see if we can target one single site of a single protein inside a human body with a small molecule. Because if we can do that, that could lead to a disruptive technological platform for precision therapeutics, because we will be able to then achieve the precision with the small molecules that have been possible only through biologics so far. But before we can achieve something like this with complex systems like humans or tissues or even live cells, we would first need the knowledge and methods that would allow us to precisely engineer isolated proteins. And once we have such methods, we can translate it back systematically to the more complex systems. But then this is the time when you should probably think about a very simple question, that do we not already have drugs that are highly specific in nature? So let me give you one such example to put things in perspective. Now, kinases are very special proteins that are responsible in regulating some critical signaling pathways inside humans. So it is not very surprising that this particular kinase inhibitor field has been heavily investigated in the past few decades. And as a result, just in last one decade, there have been more than 38 approved drugs which follow the kinase inhibition pathways. Now, if you look into some of the best protein kinase inhibitors that are available out there, they go for at least 30 to 100 identified targets. And then there are multiple unknown ones along with that. And this is the reason why you would notice that the patients who administer these drugs typically face multiple side effects. So if we are successful in our goals in the long run, we might have a possibility to address some of these questions. So in this direction, our first step was to ask a very simple question, that let's see if we can even develop chemical methods that can lead to precise single-site modification of native or unengineered proteins. So we started by looking at proteins as a multifunctional organic compound, which is full of nucleophilic residues. So if you look into one single part of it, if we want to do modification of this protein at a single site, we will in general add an electrophilic system because of the presence of all these nucleophilic residues. So this electrophile will have to first figure out how to target one type of functional group, let's say primary amine, in presence of multiple other functional groups. And once we have such a chemoselective electrophile, it would then realize that, oh, by the way, there are multiple copies of each functional group. So it has to also distinguish one functional group out of its multiple copies. And that is what I'm going to term as site selectivity in the latter part of my talk. So to sum it up, single site modification of a protein by an electrophile would require it to address chemoselectivity and site selectivity simultaneously. But let's say this primary amine is the relatively more reactive and highly solvent accessible site, and this reacts first. But then we want to also find out a way to modify this primary amine, or in another method, this primary amine. So can we go beyond the reactivity order that is defined by the protein? Can such methods be even possible? Can such modularity be achieved? So the terminology modularity will be utilized for explaining these type of selectivity factors. So with this background, we move forward and ask the very first question, that let's see if chemical methods are capable of identifying the reactivity hotspots that are present in the protein. But before we can answer this question, it was important for us to kind of address the question of concentration disparity. What that really means is that if you look into the regular textbooks or Vogel in your laboratory, the methods that are typically developed would be in the millimolar to molar concentration range. Now, for working with proteins, you will have to bring down the concentration to low micromolar range. Now that is something that is going to have a serious impact on the rate of reaction, and that is what exactly is observed. So the bioconjugate chemists solve this problem by just taking large excess of electrophile, and it does take care of the kinetics, but then it comes at a cost of selectivity. So we came up with a very simple idea that if we can design reagents that have two electrophilic centers such that the nucleophile from protein attacks on one of them in an intermolecular rapid reaction and generate another nucleophilic center that can then give me an intramolecular reaction to give the final product. And in this process, if we can shift the rate determining the step from an intermolecular reaction to an intramolecular reaction, then we should be able to decimate the role of concentration to a decent extent. We were able to realize this hypothesis with the help of N-hydroxythalamide. So in this simple example, you can see that the primary amine is going to attack on the carbonyl first, generate a latent nucleophile in form of amide, which then can attack on another carbonyl, and then you can control the electrophilicity of this carbonyl by the choice of X over here. And this works pretty well in the optimized conditions. So we took this forward and applied it to protein. And we were excited to find out that just one equivalent of N-hydroxythalamide was sufficient to give more than 50% conversion within 8 hours for this particular protein at a low concentration. Now, the important thing was that the modification happened only at a single site, which turned out to be the N-terminus alpha-amine. So that was one of the reactivity hotspots, and it proves that the chemical methods can harness the potential reactivity hotspots in a chemical reaction and target them. So with this, we moved on to the next question, and we wanted to see if we can differentiate the reactivity of a residue from its multiple copies. And for various reasons, we wanted to test this idea with lysines. But just now we noticed that the N-terminus alpha-amine would offer a higher reactivity than the epsilon amine of lysine under physiological conditions at pH around 7. So to even address this question, we had to first find out a way to block the N-terminus alpha-amine, even if transiently. And after some investigation, we found that we can design certain aldehydes that would react with protein to form imine at all the solvent accessible primary amines. Now, the interesting thing is that the imine at the N-terminus can then react with the penultimate amide bond intramolecularly to form imidazolidinone. And even though it is a reversible reaction, it is good enough to block this imine from further reacting towards an incoming nucleophile. So that creates an opportunity for testing out the idea whether one imine from lysine can be distinguished from the others. And it does work out that way, because when we take copper acetylide, we were able to target single lysine residue without much of a problem with certain optimizations. But if you want a metal-free variant, the phosphomanic reaction with triethyl phosphide did a great job, and it just works very smoothly with a structurally diverse set of proteins. And both these methods allow a lot of flexibility in terms of late stage modification and installation of diverse probes. Now, the targeting of the reactivity hotspot is not limited only to the lysine residues. We can design electrophiles or select electrophiles systematically. And in this particular case, a structurally minimalistic electrophile, that is cyclohexenone, allows us to target histidine very specifically in various proteins. And the Michael addition would give you a very simple idea. But the interesting thing is that the carbonyl that you're left with can then be utilized for installation of various probes of interest. Besides, you can utilize this electrophile for targeting the his tag in a recombinant protein. So for example, if I recombinantly express UBC9 with a his tag and then take the cell lysate, after mixing cyclohexenone with it, it will specifically target the his tag. And once you have that solution, you can take this, mix it with a resin that is hydroxylamine functionalized, and it will just simply form oxime here and capture all the his tag proteins. So now you can simply do washing, and then everything unbound will be taken off. And then you can release the protein of interest by simply treating it with imidazole through a retromichael addition and collect it in pure form. So that gives a very simple way of doing metal-free protein purification with a pre-established his tag that is typically used in most of the biochemistry laboratories for expressing protein and purifying them. So now so far, whatever methods I discussed, we were always talking about targeting the reactivity hotspots, which is typically a site that exhibits high solvent accessibility and reactivity at the same time. And at this stage, we really wanted to see if we can go beyond the reactivity order defined by the proteins and develop modular technologies. Because the potential opportunities that can be accessed with such methods, they really provide a great driving force for us, especially when we would be moving towards the precision therapeutics. But very much like the dark side of the moon, such a technology required us to address a combination of challenges. So we came up with a theory that we have been practicing for a while in our laboratory, that we can take such systems where multiple selectivity challenges are there, but then we can disintegrate them with the help of a multi-step chemical reaction that would allow us to solve them separately, and then we can integrate the solution, or the chemical reaction itself would allow us to integrate the solution. So let me just show you one example of how this could be realized in a real system. So the first example of N-terminus modification that I discussed, we added an electrophile where in one reaction it had to take care of both chemoselectivity and site-selectivity for single site modification, right? But in the second case, where we were adding an aldehyde to generate imine, this step was chemoselective. So the incoming nucleophile, whether it was copper acetylide or triethyl phosphide, that only had to worry about site selectivity. So the chemoselectivity and site selectivity are now differentiated in two steps, and depending on what goes wrong, you can tune or you know what to tune at the end of the day. So we took this forward, and we thought if it is possible to take some reactive intermediates like this and kind of generate another reactive intermediate, because if we can add such an additional step, sorry, it would allow us to test the idea of looking into residue specificity and if such transformations are even possible. So let me give you one example that worked out for us amongst various that we tried. In this case, we were working with the simple glycine, and if we treat this with this very special aldehyde that has hydrogen bond acceptor in the proximity, it leads to the formation of amino alcohol product. Now, the way it works is that the glycine is going to form an amine first, and then the proximal hydrogen bond acceptor promotes the generation of a nucleophilic enol type of intermediate, which can then react with another molecule of aldehyde, giving amino alcohol, and subsequent hydrolysis would release the amino alcohol product. Now, what is really interesting is that the orbital overlap orientations would be critical in this regard. So when you put anything else but hydrogen over here, the required conformation for the activation is not adopted, and in those cases this intermediate is not even formed, so no further reaction happens. So in a nutshell, what it means is that the reaction works very well with glycine, and it does not work with any other amino acid because all of them are substituted, and this becomes a highly residue specific methodology. So we took this forward and demonstrated that we can work with a variety of proteins ranging from a small size melittin all the way to myoglobin. This becomes even more interesting because it is pretty straightforward to express recombinant proteins with N-terminus glycine. For example, in this case, sumo was one of them. And then we were able to integrate the late stage modification and purification platform with this Gly-Tag method. And let me just show you one example of how that works. So if we take this Gly-Tag reagent and make another derivative, which is kind of a symmetric system, and then treat insulin with it. So insulin has two chains. It will have two N-terminus alpha amine residues. One of them is N-terminus phenylalanine, and the other one is going to be N-terminus glycine. So N-terminus glycine in this insulin would react specifically with this Gly-Tag reagent, give the amino alcohol product, and now you have another aldehyde motif at your leverage to capture this labeled protein on an acyl hydrazide functionalized resin. So you can recover everything that is unreacted in a very high efficiency, and it ends up giving you a single site immobilized labeled insulin. So that's how we store it in our freezers because its shelf life is pretty good. And then whenever we want, we can take it out, we can put it in different tubes and treat it with variety of probe that are functionalized with hydroxylamine. And then the transoximization reaction along with spin centrifugation gives us analytically pure single site tag protein. And that's something remarkable from these standards where the bioconjugate chemistry is now. So we took the coumarin functionalized insulin, and we also demonstrated that its binding to the insulin receptor while you target N-terminus glycine is not really affected with respect to the native insulin, and also the downstream signaling pathways in terms of phosphorylation of AKT remains largely unaffected. Now at this stage, we really wanted to see if we can take this forward and apply it to a mixture of proteins. So to begin with, we took a selected set of 7 proteins and we just mixed them manually. We made sure that only one of them would have N-terminus glycine, and then the other 6 proteins would have different N-terminus residue. And we were excited to see that in this case, when you treat it with Gly-Tag reagent, we can modify only one site of one single protein without affecting anything else. So this was all set for taking it forward to the complex mixture of proteins in a cell lysate, and that's what we did. we expressed a recombinant protein, which is SUMO1 in this case, and we were able to target that very specifically and install a fluorophore into it without really touching anything else. And that's what you see over here. So now at this stage, we were all set to see how we can take things forward. So that practically required us to develop another methodology where we wanted to find a way to dissociate this carbon-carbon bond that is formed in this amino alcohol product. But we had to do that under physiological condition. In other words, you could imagine that we wanted a retro Gly-Tag chemistry, but it turns out that when we treat this product with variety of organic bases, it's pretty robust. It really does not dissociate this bond. So eventually, we had to figure out how to change the behavior of this bond. And we were able to do that with a molecule called PLP, which would form imine with this primary amine. And then with the help of this extended conjugation, it polarizes the electron density of this carbon-carbon single bond so that you can dissociate it within physiological conditions that are pretty mild. And that releases the Gly-Tag reagent, which can be recycled and ends up giving the imine form of native protein that upon hydrolysis reverts back with the native protein. So now with both Gly-Tag and retro-Gly-Tag chemistry, we had access to a residue-specific platform that allows us to do bond formation and bond dissociation in orthogonal fashion under physiological conditions. And this is something that serves as a critical pillar for us to take our methods forward for precision therapeutics. But I'll talk about that story at some other occasion. Today, let me tell you a little bit about how we can take this combination of methods and translate that for development of single amino acid tag that can empower metal-free protein purification. So what we did is that we expressed a protein with N-terminus glycine or a few, and then we were able to translate this Gly-Tag reagent onto a solid phase. So if you mix the cell lysate with this Gly-Tag reagent, it can simply capture the protein of interest, and then you do a few washings to just get rid of everything that is not reacting with it. And now you have access to this molecule. Just either you store it that way, and whenever you need it, just treat it with a buffer having PLP. And after a spin centrifugation, you have analytically pure protein in your hand. So that works pretty smoothly. And that's as simple as what it looks like in this slide. So with this in hand, let me move on to the final question that I want to discuss in today's talk. And that was about having a modular technology that can precisely label the native proteins. And we were aware that if we are going to target a single site at a time, we really cannot go beyond the reactivity order defined by the proteins. So we came up with an idea that how about we target two residues simultaneously? So for that, we required a reagent, and in this particular example, we wanted a reagent that can have two functional groups, FK and FH, and I have highlighted lysine and histidine in this particular protein fragment. So if FK can give me a very rapid reaction with lysine and reversibility on demand, whereas FH can give me a relatively slow reaction that is irreversible but chemoselective for histidine, then what will happen if I mix this reagent with the proteins? The FK is going to go and react rapidly with all these solvent accessible lysines. And now we can regulate the micro concentration of FH near a particular histidine, depending on how I design the spacer that connects the two functional groups. And it is worth noting that this particular desired pathway has to go through an intramolecular process, whereas any background reaction of FH and histidine will have to traverse through an intermolecular reaction. And that is where you have a little advantage under physiological conditions to play with. So just to show you how these things actually translate into real examples, in the case of myoglobin, we were able to selectively target histidine 36 or histidine 81 without touching anything else in this protein, simply by the way we place FK and FH in three-dimensional space with respect to each other. And this method is not limited only to myoglobin. We can take it forward to a diverse set of proteins, and it just works equally well depending on how we design the LDM reagent. Now, once the job of FK is done as linchpin, it is available for late stage modification. So you can utilize it for installation of a variety of probes of your interest, but even better would be to capture it on resin first. So you have immobilized labeled protein, and now you have the opportunity to again treat it with the hydroxylamine derivative of the probe of interest, and centrifugal spin concentration allows you to have analytically pure single site tagged proteins. Now, at this point, we were all set to see that how LDM platform would work for the synthesis of antibody-drug conjugates. But before getting into it, let's have a quick recap of the challenges that needs to be addressed with the ADCs. So I'll show you a little video, and I will try to explain that for a simplified perspective. Okay. So as we discussed, the ADCs are composed of three key components. The toxin is the first one. In this example, it's monomethyl auristatin. The antibody that you see in the background is the second component. And then you need a linker that is going to connect the two. Now, once you see all the three components, the first question that should come into your mind is that how are we going to really control the homogeneity of bioconjugation? And once that is taken care of, and the ADCs are injected into the bloodstream, the bond strength that connects the toxin to the antibody becomes a very important parameter, because if the toxin is released before the ADC reaches the tumor tissue, it will have off-target effects. And that is why the control over chemistry and the bond type, both of them becomes really, really important. So if the chemistry and the bond type is not taken care of, the off-target effects will definitely be in there. Now, eventually, the ADCs are going to reach the tumor cells, but they might or might not kill the tumor cells. And this is where the control over the conjugation side becomes very, very important. And that's critical to ensure that the ADC antigen complex eventually gets internalized. So these are the parameters that do get regulated by the site of conjugation or the choice of that site. And then once the internalization happens, the complex eventually reaches the lysosome, where the lysosomal degradation would release the toxins as you see right now. And these toxins would be then responsible for the cell death or apoptosis. So to summarize, a powerful ADC platform is going to offer homogeneity in the antibody-drug conjugate, along with a consistent drug-to-antibody ratio over the batches. And that would, again, require the chemistry to be controlled, the residue that would be involved in that chemistry, and how does that regulate the bond strength. It should allow flexibility in terms of what linkers do you want to position. And then, obviously, if you can regulate the site of conjugation, that will be the icing on the cake. So these are the requirements that we actually are addressing with the LDM platform. So let me give you one example in this perspective. So in this representative case, we took trastuzumab as the monoclonal antibody, and then we attached emtansine as the toxin. And the purpose of choosing these two, or the combination, was to make sure that we can do a head-to-head comparison with Kadcyla, which is one of the commercially available FDA-approved products from Roche. And what you will notice is that the ADC that is derived from LDM platform outperforms the Kadcyla in inhibiting the HER2 positive or antigen positive cell line. What is even more important to note is that while it inhibits very well the antigen positive cell line, it has no negative impact on the cell line that is deprived of those antigens. So this is not really, this is not just very effective, it's also quite specific to the HER2 positive cell line. Now, the thing is that in this case, the toxins are attached to the histidine residue, and you would notice that we are looking at a carbon heteroatom bond that would exhibit some sort of fragile nature under certain conditions, and that is required for certain type of highly dense tumors. But the LDM platform is not limited only to targeting histidine residue. We can take this forward, and we can also modify lysine residues with very high specificity. And in this case, we form a robust amide bond. We demonstrated that it works very well with a structurally diverse set of proteins. It allows all the flexibilities that the previous LDM does. And then I will just share with you one representative example where we synthesize the antibody-drug conjugate, where the toxin was installed at lysine number 169 and lysine number 395. So we took this very homogeneous antibody-drug conjugate, and you would notice that in this case, the inhibition of HER2 positive cancer cell line is as good as the toxin itself, and that's really remarkable. And it's definitely way higher than the Kadcyla. Besides, it just shows no negative impact at these concentrations over the HER2 negative cell lines. So I hope that kind of convinces you that the LDM platform definitely provides a very comprehensive toolkit for accessing homogeneous ADCs and empowering directed cancer chemotherapeutics. Now with that, in last few minutes, let me just take you to one of the bottlenecks of this field of bioconjugate chemistry, and that is really related to the analysis of the bioconjugates that we prepare. So in each of the experiments that we do, the first step involved in this analysis would be simply to take the MS of the reaction mixture, and if it is single site labeled or it's a mono-labeled product, then we take it forward and treat it with proteolytic enzyme for digesting it into peptides. So once the peptides are formed, we record the MS again, and that is termed as peptide mapping. Now in the peptide mapping, we select the sequences that will have the extra tag from the chemical that was used in the modification chemistry, and that sort of labeled peptide is subjected to MSMS to identify the exact site of modification and the residue of modification. So that is a sequence of steps that has to be done with all the cases. Not for this sequence to work well and for the data to be unambiguous, it is very important that we see all the peptides when we digest the protein. Now, that is something that doesn't happen really. So if you digest cytochrome C, for example, with a trypsin, it will give you a mixture of peptides, and in routine analysis, it will be far less than 100% for sure. when you are going to look into MS. So we developed something that we term as sensitivity booster and now called as Maspecter, which enhances the sensitivity of detection of peptides in the mass spectrometry. And then we can install it into all the peptides through a chemoselective transformation, and we can then enhance the detection of peptides considerably. Now, just to give you a perspective, if I have a peptide that I can detect in the mass spectrometry at low micromolar to high nanomolar concentration, after attaching the tag, I can detect the same peptide in low femtomolar to high attomolar concentration range. So it just changes the detection level by several orders. And that is something that allows us to do a far more systematic analysis, which we also extend to the antibodies and antibody-drug conjugates and so on. But not only such reagent helps us in detecting and improving the peptide mapping, it also really simplifies the MS analysis, because now we can see all the fragments that we find in the MS. So the overall analysis itself has benefited, including peptide mapping as well as the MS. Okay, now why is this so important and why I'm putting so much of a stress on this is because in general, the assumption that has been followed in the field of bioconjugation was that if you have a single site modified protein and after digestion you find one peptide that has a tag, then that is the only thing you need. Now, all the missing peptides are definitely not labeled. Okay, so the assumption was that a monolabeled peptide, if it is seen for a monolabeled protein, that's perfectly fine. There can't be any other heterogeneity. But that is not really the case. And we demonstrated that with the help of cytochrome C again, where we designed a reagent which was activated with IISER and had a carbonyl group on the R fragment. Now, we purposefully stopped the labeling with the substoichiometric, it was too equivalent, but we stopped it at 20% conversion just to make sure that we only see monolabeled cytochrome C. Now, when you digest this with chymotrypsin, the concentration of the labeled peptide was too low for being detected. But what we did is that this time we changed the sensitivity booster functionality a little bit so that we can directly install it only on the tagged peptide. So the sensitivity of the tagged peptide can be enhanced and rest everything can remain as such. And that allowed us to analyze the two site of modifications unambiguously in the peptide mapping as well as MS. Now, this is something that really raises a red flag because it tells us that monolabeled proteins can be heterogeneous. And this is something that really is it should be noted with care when the data analysis is done for such protein bioconjugates or biologics in general. So I'll stop here and summarize my talk today. I hope I was able to convince you that chemoselectivity, site-selectivity, residue specificity, and modularity now provides a very powerful platform for precisely engineering the native proteins. From last couple of years, we have been looking into how to take this forward into live cells and target a protein specifically. And I'll be glad to talk about those stories at some other occasion. And eventually, we want to see how we are going to take it forward from there in vivo. Now, all these methods and technologies that I discussed today and a few more are accessible to the scientific community through Playable Tech. And it's time to thank all those people who did the actual work. So my co-workers have been amazing in following the dream along with me, and they have done all the actual work in the laboratory. We are really thankful to all our collaborators because you would have realized that to successfully deliver some of these interdisciplinary projects, we do need people from really different fields of expertise. And we have some amazing people at IISER Bhopal and other places as well who have been in our team since the very beginning. We are very thankful to all the funding agencies for their generous support in all these years. And thank you all for your kind patience. I'll be glad to take your questions.
Thank you, Dr. Roy, for a very nice talk. So if you have any questions, please unmute yourself and you can ask. And if you have any problem with your microphone, you can also use chat box.
Can I go ahead if nobody else is asking?
Yeah, please go ahead.
Hello.
Yeah.
Hi, this is Ruchi Vishal. Can you hear me?
Yes.
Fantastic talk. I have a couple of questions. One question I have is, you said that there was a way to purify the protein without using metal. There is already existing technology like MBP, maltose binding protein, where people use those for purification, and there is also flat tag and other things which are there.
Right.
So
Right.
can you compare what you have with them and how it is better? And should I ask my questions now or one by one? What do you think?
And anything is fine. So one by
Maybe
one would be
let's do
easier.
it. Yeah.
Yeah.
One by one.
Yeah. So yeah, no, that's a great question, and it is true that there are various tags available for metal-free protein purification, but as you would have noted that none of them are actually. First of all, you will have to engineer that additional tag separately. Whereas when you are having a recombinant protein where glycine is next to methionine, the methionine gets chopped off automatically through the enzymes, and that leaves you with glycine.
Yeah, but then you have to make all proteins with second residue as a glycine. So while you are making the protein, you have to engineer a glycine.
Yeah.
No?
So the question is whether you want to engineer one residue or you want to engineer complete tag with multiple residues or
No. So the problem, the advantage is there are already a lot of plasmids available. Like for example, flat tag and all. We don't have to really engineer them; they are already in there.
No, I
But
totally
the problem
agree.
that I see, the advantage I see is you only have one extra residue, and that you have to chop the tag, and you only have glycine.
I understand, and so I totally agree with that. But at the same time, see what happens is that with the pre-established plasmids, if you want to utilize this technology, you can still go with precision protease-based technologies. So in those cases, precision protease is always going to cleave off things next to the glycine. So you will still have glycine. You don't have to do anything extra.
Okay.
So this can easily, so in fact, one of the protein that we made, the sumo one, that actually went through that. So we took a pre-established plasmid and then we compared how, so we had his tag first, and then we did that purification, and then we used a precision protease, got rid of the things, leaving glycine at the end terminus after the protein of interest, and that works well as well. But it's just that in one case you will have nickel or cobalt contamination, but in the other case you won't have it.
Okay, good. Thank you. The other question I have is about your histidine specific labeling, if I am right. So what I noticed was most of the labeling was for exposed histidine. So there could be other histidines which were not exposed, because I looked at your structures. I mean, it was a glance, so I'm sure I didn't get the picture right.
So you got it completely right. All the residues all histidines that were modified in that slide, they were solvent exposed histidines.
Is it a technology where all solvent exposed histidines will get tagged, or can you differentiate between A site and B site, or is it a function of surface accessibility? And where does the surface accessibility, mark lies? Like if it is X versus Y, can I differentiate?
Yeah, so far, as you would probably realize that it's always a combination of reactivity defined by microenvironment along with solvent accessibility. So the challenge is that even though we can see it, so we always say reactivity hotspot, but that reactivity hotspot has two parameters, and we cannot deconvolute the contribution of each of that parameter. So one of them is definitely reactivity, and the other one is solvent accessibility. So you would imagine that something sitting in a hydrophobic domain would probably be far more reactive, right? Even in the case of lysine, if you look at the pKs, but then the accessibility is the dominant factor over there for the modification. So that's why we are careful defining it as reactivity hotspot, but we never say that it is the most solvent accessible or most reactive residue, because we can't deconvolute between the two. It is always interplay of these two parameters.
I will leave for others to ask a question. If I have one more question, maybe I can ask later if there is anybody else.
I have time, so I can answer all the questions.
I just feel I'm asking too many, so maybe I'll ask, let others ask, and then maybe I'll ask later a little bit.
Sure.
Anil, you can go ahead.
Okay. So, Anil, sorry.
Okay. Should I go ahead?
Yeah, sure.
Please.
Yeah. So Vishal, wonderful presentation. I like that remote functionalization using that spacer approach. And that reminded me of the same thing, I think, approach used by the CH functionalization guys also. When I heard Debu's talk, I see the same approach there also. So,
yes,
so
in fact,
it's quite similar to that approach.
Yeah,
so I don't know. I'm not saying it's good or I'm not saying it's bad. It's a very wonderful application of that approach.
Yeah. No, in fact, I remember that when we were developing LDM platform and I was looking at Debu's talk about how he wanted to go from ortho to meta and then to para.
Yes.
So this is exactly what we discussed, and thanks to Prasad Datta, this discussion happened in one of that brainstorming session in nice place in Goa.
Okay, wonderful. So just was wondering, you never get contamination of intermolecular reaction in these approaches?
We want to have that. In fact, when you say intermolecular, you mean
intermolecular between one end of the spacer reacting at one and the H reacts into another one and really makes it mess there.
So we really want to have that. In fact, that is something that will give us a toolbox for analyzing the interacting partners, which is interactome for a given protein. But what we realize is that the length of these spacers that we have been using, unless I'm taking two proteins that are having two binding domains that do interact very well, and I have a linchpin here that does modification here. The problem is when two independent proteins are coming close to each other, their surfaces are still a bit away. So if you do not have a spacer of sufficient length, they don't come very close to each other. And that is why the intermolecular reactions are not observed. And in fact, in some of the cases, we wanted that. So we are designing reagents where we want to do protein-directed protein modification. In fact, apart from that, protein-protein conjugation as well.
Okay. I was more keen on the homodimer side, like if you have a single protein where both the functionalities are there, but the spacer chooses one on one protein and another on another. So you get homodimer.
Yeah, so again, it will be the same thing, whether we are trying to make heterodimeric form or homodimeric form, depending on whether we keep only one protein in the mixture or two protein, and depending on their self-assembly propagation at the concentration and conditions we are keeping, that will decide whether homodimer will prevail over heterodimer and so on and so forth. But yeah, all these things will fall in the same basket as we understand. But so far, our attempts with the spacers that I have shown, they are not sufficiently long enough for resulting into protein-protein conjugation, which is a great thing because now we have, we know up to which length we can easily play with where we will not have dimerization, whether homo or heterodimerization. And then we know that, okay, beyond this, we still have to explore to see in which range we will start getting that.
Because getting long spacers which are rigid will be a challenge.
No, we don't want them to be rigid. So this was an amazing story. And if I'm allowed, I can take a couple of minutes to tell you that story.
Yeah, please go ahead.
So we initially thought that the two ends of the spacer, let's say this is one end and this is the other end, two functional groups, and this is the spacer. So we initially thought that we would need a rigid one to make sure that, you know, it's all fine. But then it was not so straightforward. We made a few rigid ones. We also made a few polyproline spacers for that. And it turned out that they just don't give us any reaction beyond linchpin formation. And we knew why. because they were not just adopting to the conformation. So that is why what I did not talk about was how the spacer was actually designed. So we deliberately kept heteroatoms at repeated space, and these heteroatoms start, so after linchpin is formed, they start adopting according to the surface of the protein. So initially they are conformationally flexible, and that is what Suman simulated and told us that initially they adopt all the possible conformation. Once linchpin is formed, they start interacting with the surface of the protein, and that is kind of adoptable conformational rigidity. So in fact, there is a technology that you might have already heard of, and it's gaining a lot of attention, that is PROTACs. Now, in case of PROTACs, it's a very similar concept along what you are saying in terms of recruiting two proteins, a protein of interest and an enzyme that can recruit the cell's killing machinery. But here it's all dependent on non-covalent interaction, and the spacer design becomes very critical on whether it can recruit two proteins or not. So the lessons that they have been learning, those lessons are something that we are also trying to see if we can adopt to that knowledge and cut down our time period on being successful in getting these things done.
So it's then assisted synthesis also. It's not basically the distance, it's an assistance from the protein surface helps you also.
Yes, that helps protein. So adaptability according to the protein surface is critical. Otherwise, having something general would not be possible.
Oh, thank you. Wonderful.
Yeah. So I have a question, Vishal.
Yes, Rajesh.
Yeah. So this is from the first part of your talk. You talked about this labeling proteins. It's some aromatic moieties, and the protein has to have N-terminus glycine.
N-terminus lysine or N-terminus
glycine,
right?
Yeah,
yes. Yes.
So now, if I have two proteins, both are having N-terminus glycine, but they have different sequences, and you want to label them, what will happen?
Yeah.
Does it matter?
No, it is an amazing question, because it kind of is the basis of one of our projects that is under evaluation and something that we are doing already from last 6 months. So you would imagine that for precision therapeutics, if we are targeting something like kinases, they have N-terminus glycine, but then they have a lot of family members, cousins and brothers who also have N-terminus glycine. So how do we differentiate one N-glycine from the other? So in our model experiments, we found that, the rate of reactions are different, but by the time you achieve 10 to 20% conversion with one of them, the second one will also start participating. So for the practical purposes, you can get some differentiation, but not exclusive differentiation that you would like to have.
Because in your scheme, I could not find another N-terminus protein with a different sequence. You had like 6 others or 5 others, but they did not have this N-terminus glycine.
Right. So that was
ideally you should have 2 proteins with N-terminus glycine, 2 or more proteins, 2 with different sequences.
So that was deliberately picked. You rightly pointed it out. So in the mixture that we took initially 7 proteins, only one of them had N-terminus glycine. And even in the cell lysate, there were other proteins with N-terminus glycine, but then the recombinant protein was overexpressed. Right. So you had one in much higher concentration and then control the extent of modification. But this is upcoming. In another couple of years, hopefully you will see another technology, which is actually integrated form of LDM and Gly-Tag technology, which allows us to distinguish N-terminus glycine of one protein from the other. In the model peptides we have tried so far, we still have to try it with proteins.
Thank you.
Thanks.
So Vishal, wonderful presentation. So can you hear me? This Pradeep?
Yes, I can hear you.
Yeah, wonderful presentation. So I have a question related to the last part where you are increasing the sensitivity of the mass spec analysis. So can you, I missed something. So can you tell how this sensitivity is enhanced by tagging these fragments?
Okay, so no magics. You want so there are 2-3 elements that are important. In fact, 2 are more important than anything else. One of them is that you always want a positively charged center.
Right.
So that is something that we do with a few motifs that involves guanidinium and a few more heterocyclic motifs. Apart from that, what you also have to worry about is convenient desolvation. So that we do with a hydrophobic chain. So these two factors, one, that will make sure that, you know, whatever you have, everything is always going to have ionized form, which is having very high ionization affinity. And the second part is something that helps in desolvation. So you make sure that everything that goes in is detectable by MS. And that's the only trick.
But then it requires the that all the fragments need to be uniformly labeled. So how do you ensure that?
So what happens is that it would label all. So that is why we keep NHS-IISER at one end of that reagent. So it chemoselectively labels the N-terminus alphamine. So 2.2 to 2.5 equivalent per peptide is what we use. And then it chemoselectively goes to N-terminus alphamine. But if you have C-terminus lysine, then it prefers to go at C-terminus lysine and not at N-terminus alphamine. But in either case, you know whether it will be at N-terminus alphamine or C-terminus lysine, nowhere else. So now what that does is that it practically, and when you're using 2 to 2.5 equivalents, so far what we have observed is that it is pretty homogeneous labeling. But yes, you can argue that it won't be, if it is 100% for one, it need not be 100% for the second as well. Have I answered your question?
Yeah, so I got the idea, so that's fine. Yeah.
But if you want to use some of these, I'll be glad to send some samples. We are looking for volunteers who want to utilize some of the sensitivity booster.
Yeah, definitely. Thank you.
So,
Vishal, I have another question. So you were saying that you were trying to do intramolecular reactions because of concentration related issues that you were mentioning. But I thought the main concentration related issue was having enough protein and not having intramolecular intermolecular reactivity problem unless if we go to different groups. So in that regard, do you have any comments?
So I guess you are talking about this initial example where so see, the thing is that protein you would like to have in 20 micromolar to 50 micromolar range. Going above 100 micromolar,
yeah, I understand that. I understand.
Right,
the protein is a limiting factor.
Yes.
Of the reagent, right?
Right. So now that is why, you know, equimolar that we keep up, you know, if we think as organic chemist and then we think of how with this small molecule, when we say equimolar, it means 1 millimole to 1 millimole. That doesn't really apply in the case of proteins. Because then the localization, the differential localization and all those starts coming into play. So you never know what is the exact effective concentration at a certain place. So effective concentration becomes an added parameter.
I thought that if you have less amount of dye, in this case, whatever your reagent,
yes.
And the reagent has a strong preference for one group at low concentrations, having you know, side reactions would be minimal. So that is what is most desired.
Yes. So what happens though with most of the electrophiles, with an exception of maybe few, like NHS-IISER or melimide, you would notice that for most of the electrophiles that show common reactivity or good reactivity with the small molecules at millimolar concentration range, they would just not give you any reaction if you will be using substoichiometric amounts or even equimolar. So if I take, let's say, alkylating agent and I put one equivalent of that into this protein RNase A, it will not give me any reaction for several hours. And so that's one common problem that you'll find, that most of such electrophilic systems at very low concentrations, at 20 to 50 micromolar concentration range, they will simply not give reaction. So that is what I was talking about when I said the kinetics is severely affected at very low concentrations. Have I answered it? Or
kind of, it's okay. Yeah, I understand the problem a little bit more.
Yeah. And you would imagine that since our whole ambition is to eventually go in cellulo and in vivo, we really did not wanted to deviate from what we are going to face under those circumstances. So we wanted to stay close to that. Because anyway, there will be a lot of changes due to compartmentalization of cells and later tissues. So we did not wanted to add more variables to it to make it further complicated at this stage.
Hi, Vishal. I have one or two questions. Hi. What you started with that premise that okay, kinase inhibitors are not specific and those things. So
yeah,
essentially, and what if I understood your LDM thing correctly, is mostly like kind of a ligand molecules that are targeting some of the receptors that are highly expressed in the cancer cell lines or something like that. Okay.
No.
No.
So,
okay. Okay.
So
then I.
Yeah. So ligand directed modification is another technique. And I would
say, for example, you did it with say trastuzumab or say insulin or those proteins that you talked about. Mostly insulin activates insulin receptors.
Right.
Trastuzumab is an receptor inhibitor. So and obviously need to be those needs to be internalized in a proper manner. Okay.
Yes.
So but the kinase inhibitors that we talked about is mostly global. Say, for example, I have AKT inhibitor also, but AKT is not inside. Mostly inside the cells, not outside. So in a sense, what I didn't understand, say, I can understand that you can specifically modify proteins and you can do that job. How you're going to do it? So some kinase which is already inside the cell, not outside, and you want to do something like that. Say, for example, yeah.
So, the story that I discussed today was mostly centered around directed therapeutics with ADC and the technologies, how they are leading towards that. What I did not discuss is, so I just kept this as kind of a future target that we are trying to achieve. So what you would need in case of, let's say, kinase inhibitors is, so for example, let's say if we are targeting SRC kinase, and we just want to target that specifically and nothing else, then so that is where we are, that's one system that we are already working on. And now you have to target one sequence, and depending on the sequence, it show specificity. So the specificity is not something that is derived from antibody in terms of targeting the SRC. But you can very well argue that after we have developed that technology, at least still in cellular and even subcutaneous in vivo model, then after mice, if we have to take it to the tissues that are internally placed, then it would be little different. Then we would probably need a help of another delivery agent, and that would be very well antibody. So that is the segment where this antibody conjugation technique will be kind of coupled with the precision technique. So antibody will help us taking something to a certain tissue, and then after that, the precision of that particular reagent has to play the role. So
good luck.
I totally.
It's really challenging. Yeah.
It is, right? So, but the fun thing is that, slowly while we are working on these things, we are taking care of different pillars that would eventually be required for that. And the best part is that even if we are able to address one of those things, it will still be better than what is currently existing. So we will have time to gradually prove ourselves over a period of years.
Very nice. Thank you.
Thanks.
Any other questions? Okay, if not, let's thank Dr. Rai. Thank you, Dr. Rai, for accepting our invitation and also presenting a nice talk, nice work. And let me also thank all the participants who joined today. and see you all in the next webinar. Thank you.
Thank you all. Have a great weekend.